Hi, thanks for joining us. We're pretty excited to present our work that we did on Humboldt Bay Coho salmon. My name is Josh Cahill. I work for the Eureka tribe environmental department. I'm a former grad student in Dr. Darren Ward's lab at Humboldt State.
And I'm Kate Stonecypher. I currently work for Cal Poly Humboldt, and I'm a current grad student at Cal Poly Humboldt. And we are presenting our combined theses projects today, which we titled Ecology of Recovery, Research and Monitoring Floodplain Habitat Restoration for Threatened Coho Salmon. So we were working with the Southern Oregon, Northern California Coast Evolutionary Significant Unit of Coho Salmon. This ESU was listed as threatened under the Endangered Species Act in 1997. The lack of floodplain habitat has been identified as a key limiting stress for most coastal populations and about half of interior populations for this ESU. These areas historically provided velocity refugia for juvenile coho from high winter flows, and the loss of these habitats has impacted their survival within their first year of life. Pacific salmonids are important culturally, economically, as well as ecologically, as many of you know. Culturally, they provide sustenance and a symbol of renewal for local indigenous tribes. They're a huge part of their cultural and spiritual identities along the Pacific coast. Economically, in the West Coast, the commercial and recreational fishery in 2022 was worth almost $80 million. And in the Port of Eureka alone, these fisheries represented almost $500,000. Ecologically, salmon serve a critical role in providing marine-derived nutrients to freshwater and terrestrial ecosystems and support multiple trophic interactions with organisms throughout the forest and different freshwater environments.
So we wanted to acknowledge that all the research that we conducted took place on Wiyot territory, and that we recognize the stewardship that the Wiyot people have provided and continue to provide for the landscape and for the waterways. So Wigi, or Humboldt Bay, drains about a 580 square kilometer area. Within this area, the Eureka coastal plain lies in there. And historically, it was comprised of a very diverse and complex habitat that had a lot of terrestrial, freshwater, marine, and estuary environments. And the majority of this was diked and drained for agricultural development. So there's, on the slide, there's some of the different habitat types that once existed in Humboldt Bay. And this image on the screen shows the loss of wetland habitat in the Humboldt Bay region. So panel A is from the 1890s. You can see a nearly contiguous border of wetland habitat that used to surround the bay, and panel B depicts what was left in the 1970s, with most of the habitat loss occurring before 1910. So that's about 90% loss in habitat. So pictured here is Tolwa Island in Wigi, and you can see from this photograph that the habitat is very complex. There's meandering channels that vary in depth. There are alcoves and ponds. And this is what the Eureka coastal plain was thought to look like before European contact. So in the literature, it's been identified that increased stream velocities due to simplifying channels is a direct threat to juvenile coho salmon survival. And also identified in the literature is that a complex habitat across a large spatial scale really improves ecosystem resilience and populations resilience. So what this means for Humboldt Bay coho salmon is that there's less refuge habitat to get out of high winter flows. There's decreased habitat for the whole smoltification process they go through before transitioning into the ocean. And as you'll find out pretty soon with some of Kate's slides, different habitat types offer different prey items available to the fish. So this decreases foraging opportunities for the fish as well, and leads to the decline of a population.
So there's been a significant investment in floodplain restoration across the Pacific coast and here in Humboldt Bay. Billions are spent annually on stream restoration in the United States. Historically, very few of these projects were monitored, and even fewer were evaluated for their efficacy. So the core of our work is to understand how these projects are contributing to recovery criteria for listed coho. How do factors such as their abundance or survival compare to the broader population of fish within these sites and without? So we combined our work for the purpose of this presentation. Very similar objectives. Josh finished his thesis last year in 2023, and I'll finish mine here this year in 2024. Josh's objectives were to determine if coho salmon were using these newly restored sites within Humboldt Bay tributaries and assess their movement between main stem and restored off-channel habitats, as well as to estimate their survival and growth within the off-channel pond and main stem habitats. And then my work came after Josh's and built on that foundation by investigating diet, prey availability, and growth of coho that are using these restored off-channel habitats as well as main stem habitats, and to estimate abundance and survival of fish using restored sites and compare that to the broader population. So on this map, we have all of our sites in the Humboldt Bay watershed here, 4 main tributaries north to south: Jacoby Creek, Freshwater Creek, Elk River, and Salmon Creek. Josh's estuarine sites are shown with the blue squares, and his freshwater sites are shown with the green triangles. Ross Taylor and Associates collected data from Howard Heights for Josh's project, and then my sites are shown with the pink circles. I had Howard Heights as well as Upper Jacoby Creek, so we had overlap at those locations. And then I was also sampling Cochrane Creek for my project, which is my estuarine site. And then I just want to highlight the Freshwater Creek Life Cycle Monitoring Station here, which is operated by the California Department of Fish and Wildlife Arcata, because they provided a lot of data and support for both of our projects. So our methods, we're using pit telemetry, going out and capturing juvenile coho with pulse seines, marking them with pit tags. We use mark recapture analysis to estimate abundance, movement, growth, and survival. And for the diet portion of my study, I use non-lethal gastric lavage to collect diet contents from juvenile coho in both off-channel pond and main stem habitats.
All right, so I sampled 6 locations on in different tributaries to Humboldt Bay, and Ross Taylor was kind enough to sample a 7th site that I could. He shared his data so I could include it in my analyses. And this figure is before any like real statistical analysis was done. I was looking at the raw data and sorted fish because I wanted to see what fish we were tagging. What was the outcome? Did we see them again? Did they move around, or did they express site fidelity and stay put for most of the season? A couple of quick trends before talking about the bar plot is you can see that capture and use of these sites by coho salmon is not equal across all sites, but we caught salmon in all of the sites. 2, we caught more fish in 2019 than 2020. And 3, fish moved into restoration sites from the creek habitat earlier in the season in 2019 than in 2020. As far as this bar chart is concerned, the pink areas depict fish that had moved, that were captured and tagged at a location, and then they were detected again later on somewhere else in the system. The green portion of the bar plots are fish that were tagged that were never seen again. And the blue portion are fish that were captured and tagged and then detected again in the same site later on. So I wanted to also look at movement of the fish when fish moved from creek habitats into estuary habitats, because a lot of older literature and older survival estimates of coho salmon looked at early immigrant fish into marine and estuary habitats, and they were counted as mortalities. And we're moving away from that trend now, but I wanted to see what it was like in the systems that I was studying. So I performed a multi-state mark recapture model, and I have a hydrograph at the top of the figure for reference. And the main takeaways from this slide is that we saw patterns of movement in both watersheds, Jacoby Creek and Freshwater Creek, and across cohorts. So 2019 and 2020, we see this pulse of movement in the early winter, and then another pulse of movement in the spring outmigration period. But I wanted to point out that the magnitude of this pattern is different between watersheds and between years. So for example, if we look at Jacoby Creek, for the early winter movement period, we have a really high probability of seeing fish moving from the creek into the estuary habitat. And if we look at Freshwater Creek, and mind you, these two watersheds are right next to each other, so they're experiencing similar discharge and similar atmospheric conditions. This pulse of movement is much smaller in Freshwater Creek. And then when we look at 2020, the trend is reversed. Freshwater Creek has a really high probability. So excuse me, I think that the results of movement really highlights the importance of considering habitat diversity, since we had fish expressing different life histories under the same conditions. And with the same mark recapture model, we were able to estimate apparent survival in creek rearing fish versus estuary rearing fish. Just to recap, older survival models typically counted early migrants, early immigrants as mortalities. So I wanted to see what the survival was in estuary restoration sites and estuary habitat. So if you look at the Jacoby Creek estimates between both years, the estuary and creek survival, apparent survival estimates are very similar. And when you look at Freshwater Creek in comparison, the estimates for estuary rearing fish is always lower. And I don't think that my model is suggesting that Jacoby Creek estuary is better rearing habitat for coho salmon. I think that this is actually showing that sample design and monitoring is much different between the two systems. So at the time that I did my research, the only habitat, estuarine habitat, that's connected year-round in Jacoby Creek was monitored with a pit tag antenna array that Colin from CDFW was maintaining. And so we could track fish going in and out of that estuary habitat. Whereas with Freshwater Creek, there's miles of estuary habitat that fish can move in and out of without ever being detected. So I think that again, this slide highlights the need for better monitoring so we can understand the systems that we're studying better.
These are just some Lincoln Peterson abundance estimates from my restored off-channel pond site. As I mentioned, I'm not done yet, so I don't have my fancy robust design survival modeling to show you. But as you can see, there's some pretty big differences between sites. I was going out twice a month doing paired events from January through May of 2022, and overnight installing a block net so that I could estimate abundance within these habitats. And some pretty big differences at Cochrane Creek. Abundance was pretty low throughout my sampling period. It was typically around 6 fish. At Jacoby Creek, abundance really peaked in February at almost 500 individuals. That's a lot higher than my other 2 sites. And then just seasonal differences in detection. By May, fish had emigrated from both Jacoby Creek and Cochrane Creek, but were still present at Howard Heights within the Freshwater Creek watershed. So these are both of our growth models side by side, Josh's growth model in length on the left and my growth model in millimeters per day on the right. They're differently structured models. Josh's is a generalized linear model and mine is a nonlinear model, but they show some consistent results, which is pretty cool. And that is that growth is higher at the estuarine sites than at the freshwater sites. For example, at the golf course, which is Martin Slough, growth is a lot higher than at Upper Jacoby Creek, which is a freshwater site. I kind of saw the same thing at Cochrane Creek. Growth is a lot higher than at Freshwater Creek or Jacoby Creek off-channel ponds. And a main take home from Josh's growth model is that fish move into these restored estuarine habitats at a smaller size, but grow larger over a shorter period of time than fish in the freshwater floodplain habitats upstream. And then a main take home from my growth model is that fish were growing faster in both Cochrane Creek and Freshwater Creek off-channel ponds, but not at Upper Jacoby Creek off-channel pond. I also noted some pretty big differences in diet composition across sites. This is kind of a vastly oversimplified figure for what is a pretty complex data set, but some of the nuances are the same for these samples. Notably, mysid shrimp, which are an intertidal species of possum shrimp, were ever present in the diets in the estuarine sites and could be a driving factor behind the increased growth there. Terrestrials are an important contribution across a lot of sites and locations. And then zooplankton, like these Daphnia, were present in all off-channel pond sites, but they were only a noticeable contribution at this upper Jacoby Creek pond. That was also the site with the lowest growth as well as the highest abundance. So those two factors could be a mechanism that explains that lower growth compared to the other off-channel pond sites.
We just wanted to point out too that we caught some anomalies where, some fish that we're not sure what life history that we can assign to them in the classic coho salmon life history sense. I caught this fish in Martin Slough on the Eureka Golf Course in January of 2020. And more recently, my lab mates, Kate and Colton, have captured these fish at South Jacoby and at Janes Creek. And all these fish are about 300 millimeters, which are monstrous fish for finding in streams and estuaries, and Colton pulled this scale from this fish captured in Janes Creek, and it's really interesting to look at the uniformity of the annuli here, which suggests that this fish probably did not move out into the marine environment. And so yeah, like I said, it's kind of difficult to try to say precisely where this fish has been living and what it's been doing. So yeah, this is just another instance to highlight the importance of monitoring and of habitat diversity to allow different life histories to express. And so with that, we just want to say thanks to our families for being understanding, going through this whole process, our advisors, Dr. Darren Ward and Allison O'Dowd, the people that were kind enough to contribute data and allow us to contribute to their longstanding work, like Seth, Colin, and Mike from CDFW, Ross, Bob from NOAA, and our funders, all the people that helped us in the field. It wouldn't have happened. It took a village for sure. So thanks.